All right, so today, happy Monday, we're going to be looking at how the cell cycle is regulated. And I know I made a few comments that this is going to be ultimately what we were going to talk about today on Friday when we looked at mitosis and cell division itself. But this is actually a pretty important topic because as we're going to use as an example, it has very important ramifications for the development of cancer and the treatment of cancer because cancer is an example of when the cell cycle becomes dysregulated. So cells start to go through mitosis and divide when they're not supposed to, or they end up living longer than they're supposed to. So, in all multicellular organisms, they need to kind of coordinate cell division across different organs and across different tissues. So, it's really important for normal growth and development and maintenance that when a cell dies, it's replaced, and when new structures and new tissues are supposed to be being made for a growing organism, that they're being made, and that this all happens in a coordinated manner. So, uh, a bunch of cells aren't made without the need, and when there is a need, the cells are made on time. This becomes especially intricate when you discover that not all cells have the same cell cycle. They're not going through that cycle in the same amount of time. Like last time we talked about the G0 phase and how some cells are in that for as long as the organism is alive, and others may go in and out, and some go through mitosis in the cell cycle really rapidly. So coordinating the timing is everything as well. So a little bit more on that. Frequency of cell division varies by the type of cell. So say we had two cells that are in the cell cycle, they're not going off in a G0 phase. The time it takes for both of those cells to go through the whole step and back to the beginning won't be the same. For an example, in a developing embryo, the cell cycle will take less than 20 minutes. All right, so think about that ball of cells that's rapidly dividing to make a new organism. Those divisions happen really, really rapidly because we're in the most rapid phase of growth of that organism's life. Skin cells divide really frequently throughout like our lifetime, so that they'll go through one cycle of the cell cycle every 12 to 24 hours because your skin is being always replaced to provide a nice barrier to the outside world. Your liver cells, like we used as an example on Friday, always have the ability to divide, but they keep in reserve. They might divide once every year or two just to replace cells that are naturally dying off. And mature nerve cells, like in your brain and muscle cells, don't divide after they're mature. They're permanently in that G0 phase, right? G0 phase. And it might come as a surprise to you that muscle cells don't divide after maturity. Okay, that might be kind of weird, but think about, for example, if you have like heart conditions, like a heart attack where part of the coronary blood vessels get blocked and oxygen doesn't come back, isn't able to get through and supply that muscle with oxygen. The real dangerous part is that, even if you'll recover, you'll permanently have an area of your heart that might have dead parts, and those, can't ever grow back. And that's kind of why you'll see a lot of long-term chronic damage from somebody that has had a heart attack. That's why it's so important. All right, so to control all of these different rates, there are two irreversible points in the cell cycle, and we call them checkpoints. And one is where the replication of genetic material takes place. And if we remember, that is in the S phase. And the other is when sister chromatids separate in metaphase. Both of these are called checkpoints, where the cell stops in the cell cycle to make sure everything is okay, and that our genetic material is replicated, and that these sister chromatids separated in will be separating like an anaphase, and that things can go on and proceed normally. So we don't end up with cells that aren't with their full complement of DNA. So it's at these checkpoints that if everything's okay, the cell will continue to go through the cell cycle. But if things aren't okay, it might stop everything right there. All right, so a little bit more about the checkpoint control system. So for the checkpoints, the cell cycle is controlled by kind of stop and go chemical signals at these critical checkpoints, right? Um, these signals indicate if very important processes have been completed correctly that'll be necessary to continue through the cell cycle. And if not, it will stop the cell and its progression through the cell cycle or mitosis until those processes can be completed. So if you look here, there are three important checkpoints that we're going to talk about. There's more, but these are the most important ones we're going to talk about. The G1 checkpoint, that happens in the middle of G1, the G2 checkpoint, which takes place right after G2, and the M checkpoint that takes place around metaphase of mitosis. Yep, there's three major ones that we just kind of talked about, just mentioned. The G1S checkpoint. This one really needs to answer the question, can DNA synthesis begin? Do we have all the resources and the necessary material to synthesize DNA? If no, the cell will stop proceeding through the cell cycle there until those conditions have been met. If yes, the cell's going to be free to move on through G1 into the S phase. The next is the G2 slash M checkpoint. At this point, the cell is making sure that DNA has been synthesized correctly, because if the cell would end up going into the M phase without the DNA being synthesized, being duplicated, you would end up having daughter cells that would have different numbers of chromosomes, different amount of DNA than they should. This is kind of like the last check, no turning back commitment to mitosis if the G2M checkpoint is passed. And then last is the spindle checkpoint, or the M checkpoint. In here, we want to make sure all chromosomes are attached to those spindle fibers from the centrioles during metaphase before we begin anaphase. This is ensuring that sister chromatids can separate from one another again, so that each daughter cell has the correct number of chromosomes. All right, so here a little bit more about the first one. That G1S checkpoint is kind of the most critical. It's the primary decision point within the cell cycle, or a restriction point. If things pass this, it's more likely that they will complete the whole cell cycle. Internal signals like the cell's size or growth, the amount of nutrients it has, and its external conditions really influence this G1S checkpoint. If cells do not receive the signal to divide at the G1S checkpoint, it will exit the cell cycle and it'll switch to G0, that waiting phase, until it receives the external signals, the growth factors, um, the nutrients, and things like that. So it's really the restriction point of the cell cycle. And then we kind of already talked about the G0 phase last time. This is just kind of review, so I'm going to put this up for a second. So if you still need to review some of this from last time, feel free to pause this. But otherwise, I'm going to kind of skip over this for now since it was part of the last video as well. So then the question becomes, okay, we know how we can regulate this cycle to make sure that the cells are prepared to divide when they do, but how do they know when to divide? What are those signals we're talking about? So, as some examples, there are chemical signals in the cytoplasm that will give the cell cues when all of those conditions are met to pass the checkpoint. They usually are in the form of proteins like activators, which will speed up the cell cycle, or inhibitors, which will slow down or stop it. One of the reasons that we know this is when an experiment when a cell that was in the mitosis phase and a cell that was in the G0 phase were joined together, both of them started to go through mitosis. And so that is kind of evidence that chemicals or signal molecules from the M cell ended up stimulating the cell that was in G1 to divide. So the molecules that are kind of those signalers, those go ahead and divide signals are these proteins that promote cell growth and division. They are an internal signal, so they originate within the cell. And we sometimes call them promoting factors because they promote cell division. There are others that are called external signals that come from outside of the cell that we call growth factors, right? Causing the organism to grow, divide, make more cells. These internal promoting factors and external growth factors are the primary mechanisms to control cell division. And they do that in a similar way to the signal transduction pathway we talked about last Thursday, which is via phosphorylation, adding a phosphate group. So just as we saw in that cascade, we'll have active and inactive versions of these signaling molecules. And when we add a phosphate group to them, or phosphorylate them, they will become active and promote cell division. And when there is no phosphate group on them, they'll be deactivated, and they'll no longer signal or promote cell division. So cell cycle signals can kind of be broken down into these three molecules that will really help us control. So cyclins, which are regulatory proteins, their level of activation kind of goes in a pattern throughout the cell. So as an example, during the moments right before mitosis, their levels will be really, really high. And right after mitosis, their levels will be really, really low. So they're called cyclins because their activity is cycling throughout the cell. CDKs, or what's short for cyclin-dependent kinases, they're the ones that phosphorylate cellular proteins, mainly cyclin, and they activate or inactivate them. And so cyclin-dependent kinases will bind to cyclins to form what we call a cyclin-dependent kinase cyclin complex. Here is one that is active. It is active only when cyclin has bound to it and it is phosphorylated. This complex triggers the passage through different stages of the cell cycle by letting the cell go past their checkpoint. right? And in today's assignment, you're going to look a lot more closely at this process. There's going to be a model that you're going to look at that helps you understand how this interaction happens. So these are a really interesting picture, kind of telling you what we're talking about, how the cyclins cycle throughout the cell cycle. Their levels go up as mitosis approaches, and they drop back down in mitosis, and then they start rising again. That's why we call them kind of cyclins. So if we kind of put this in a picture, let's start at right after we got into mitosis. So we're going through the G1 phase, and we get to the G1 checkpoint, and we're waiting to see whether the correct growth factors have been received, the nutritional state of the cell is good, the size of the cell is good, and if that's the case, these cyclins and the cyclin-dependent kinases will bind to each other, and the cyclin-dependent kinase cyclin complex will be active and allow this checkpoint to be bypassed. And then we're going over to G2 phase, and if all of those checkpoints are completed, again, the cyclin-dependent kinase cyclins will bind together to form that complex, and we can move into mitosis. Right? The same thing with the last phase of the last checkpoint in mitosis, the spindle checkpoint. So cyclin-dependent kinases and cyclins drive the cells from one phase to the next, one checkpoint to the next. Proper regulation of the cell cycle is key to life, so that the genes for these regulatory proteins has been highly conserved. What that means is that most of life have these same genes to regulate the cell cycle that are for cyclin-dependent kinases and cyclins. They're basically the same in yeast, insects, plants, animals, and humans. They're very old, they're a very ancient evolutionary origin. There are also external signals like we talked about. So cyclins and cyclin-dependent kinases are internal signals. They originate from within the cell, but there are also external signals for cell division. So we call these growth factors. They are kind of coordinated between cells, and they're proteins that signal the release by the body cell that stimulates other cells to divide. So a cell might send these out to stimulate other cells to divide. So this is what causes what's called density-dependent inhibition, where crowded cells that are crowded together stop dividing, um, because each cell binds a bit of growth factor, and there's not enough active growth factor left to trigger cell division in any one cell. So that's what makes sure that normally when a tissue has as many cells as they need, that cell division doesn't continue and a tumor doesn't develop. And then there's also a thing called anchorage dependence, which means that in order for a cell to divide, it must be attached to something else, some other cell. So that prevents, for example, um, cells breaking off in your bloodstream and then multiplying out of control. So in this example, that's why if you put cells in a petri dish, they will grow to cover the whole petri dish, but then they'll stop. It's because of this density-dependent inhibition and this anchorage-dependent inhibition. So this is just a general picture of the growth factor signaling. You don't have to remember anything specific about it. Just notice how it models that signal transduction pathway that we talked about from a receptive phase, transductive phase, and response phase. All right. I think this is where we're going to stop today. Next time, we're going to finish up with a little bit about just reviewing this cell cycle control and then talking about how it eventually gives rise to cancer.